Okay, it's my pleasure to introduce our seminar speaker, Gaurav, and I have to pronounce it correctly, Sukhatme.
Well done.
Is that right?
Well done.
He's saying well done, he's not saying it's right. To the gentlemanly level, he's a gentleman. So Gaurav got his a PhD from USC in 1997 in Southern California, and he liked it so much there, and they liked him so much that he decided just to stay and become a Southern California boy, filling all the stereotypes that you figure you'd see in Southern California. Gaurav's had a pretty comprehensive career in robotics, touching a variety of fields. But the thing I want to point out is his service to our society, to the Robotics Society. Well, for one thing, he led a MIRI that I was part of, and that funded Glenn Wagner's work and led to other fine projects for us, so selfishly, thank you. But he also is one of the founding members of the RSS conference, which has become the premier conference in the robotics field. So for that, as a community, we thank you.
You're welcome.
We finally would like to thank him once more for taking his time, because he's got a very busy schedule, to come all the way out here to Pittsburgh, where it is sunny today, now, and give his talk. At this point, I'm going to remind everyone to turn off your cell phone. All right, please.
Thank you so much, Howie. So, thanks very much for inviting me, and thank you all for coming. I'd like to spend some time today telling you about one recent project in my lab. We pursue lots of interesting things, lots of fun projects we pursue. I picked one. I hope you'll find it interesting. We've been working with underwater robots for a while, and this is one of our recent adventures. So my title sort of may not necessarily explain a whole lot. You should view this talk largely as a problem in constrained decision making. So we're going to deploy an underwater robot in the ocean. It's going to make a set of sequential decisions, and these decisions are in the service of some overall mission. And we're going to argue that this kind of decision making problem is key to long-term autonomy for underwater vehicles. And so it's really a constrained decision making problem talk. Okay? And I also want to point out that we collaborated with a lot of people in this work, but the real work was done by Naneshwar Das, who is the PhD student who worked on this and is now a postdoc at Penn, and really moved this project forward and did the lion's share of the work on this. Okay. And so a few years ago, I became very interested in underwater robotics because I was in conversations with some friends of mine who work in marine microbiology and in oceanography, and they told me that the only place on Earth worth studying is if you're drenched in water. Everything else is pointless. And they enumerated the reasons for me as follows. Right? So life began in the water, and the biosphere is almost entirely contained in the hydrosphere. Not completely. We're exceptional, as it turns out. And a lot of habitable space on Earth is in the ocean. We don't know a whole lot about this ecosystem. And to a robotics person, this is an opportunity. Robotic exploration is changing this. So our lab became part of this journey of trying to help in this endeavor. All right, how many people here have worked with underwater robots? Okay, so for the rest of you, what can you learn from this slide about underwater robots? They're all yellow. Okay, so hopefully, with no offense, I can say that's machine learning 101 for underwater robotics. We've quickly done an extrapolation. They are indeed yellow because when you go find them, we want them to be easy to see. But the real point of the slide is to show you that there's a huge diversity of underwater robots out there in the world. There's different shapes and sizes, and some even draw power from the sun, and some are deep ocean type vehicles. Some are kind of more like toys. They have various thruster configurations and so on and so forth. It's a lot of diversity. If you want to build your own underwater robot, you can even get a yellow book. It has to be yellow, and you can build one, right? But okay, so in the underwater space, our lab has focused on 4 big areas over the past decade. I'm going to spend one slide telling you what these areas have been, and I'm going to pick one of these, and I'm going to talk to you about that project. So this picture is a view from the sky of the coastline in Southern California. So the city of LA is here. This is a small island off the coast of Southern California called Catalina Island. This is obviously the blue stuff is all ocean, and these kind of highlighted streaks that you see are actually a record of the shipping traffic entering and leaving the port of LA, right? So this is where all the stuff from China and Japan comes into the United States, and then by road winds its way through the country. It's an extremely busy port. If you deploy underwater robots in this area, one of the key challenges is that if you end up hitting one of these ships, then very often the ship is fine, but the underwater robot is history. And so one of the things we worked on quite a bit is risk-aware path planning for these kinds of very crowded environments. And the idea is to be able to listen on the transponder for ship traffic, to have priors of where the ships generally tend to be, these shipping lanes, and to be able to design techniques for vehicles to navigate in these environments so that they, generally speaking, end up not hitting one of these ships. The problem is not complicated because only of this. The problem is complicated because the water is pushing the vehicle around. And that's what makes the problem hard. So your water forecast is updated typically every 12 hours by an ocean model, and you have to reason stochastically about how you're going to get pushed around by the ocean in the hours to come. And that's what makes this path planning problem difficult. So we have a body of work on this. Also off the coast of the western United States it's a seismically very active area, as all of you know. And so there's actually a set of seismometers denoted here by these diamonds that are embedded on the ocean floor. And one of the problems we worked on is designing techniques for an underwater robot to visit these to grab data from them. So how do you design good tours for underwater vehicles to mule the data off of these seismometers and bring it back to shore? So that's been a recent thrust, and we have some interesting results on that as well. We've also worked quite a bit on active classification and inspection problems. So this curious-looking contraption is the underside of a ship. This is the propeller. This is the rudder, and this is sort of the underside of the hull. It's false colored for the following reason. The kinds of problems we're interested in here is to deploy an underwater vehicle while the ship is in harbor. And so some of you may not be old enough to remember when the USS Cole was bombed. It was moored, it was in harbor, and a bunch of people went up and stuck some explosives to its side and ripped a huge hole into this warship, basically, right? And so there's been an interest in the Navy since then of having small robots patrol the undersides of ships so that this doesn't happen. And the challenge is, you have a CAD model of the underside of the ship. The question is, in a very short period of time, how do you design tours to explore the geometry of the underside so that should someone have tampered with the hull, with maximal likelihood you're likely to detect it? Right? That's the problem position. And so in fact what the coloring here shows are regions, as you may guess, regions of high curvature, where it is possible to in fact attach something and be unobtrusive. It's relatively hard to do that on a plane. That's all that picture shows. So there's a science to how to solve the view planning problem, where if the challenge is you need to put a robot in and it needs to survey this very large geometry, but very quickly, to give this guarantee that largely speaking tampering will not go unnoticed. So we have sort of a nice line of work on that. And this last picture shows this is actually the Embari Dorado vehicle on one of its journeys in the ocean. We've been interested for a while in how to endow vehicles like this with algorithms that make them good at taking samples of water for biological analysis. And this is a project we've been working on in collaboration with some marine microbiologists. And so I would like to tell you a little more about this one today. What these projects all share in common is we tend to think of problems of path planning for underwater vehicles as being central and important towards their increased autonomy, particularly because many of the techniques that work on land don't work quite directly given the ocean, given the external forcing associated with the ocean and the lack of good forecasts for this. Okay, so the problem is as follows, right? The problem is we want to go in environments where we want to bring back samples to a lab. So there's a lander on Mars, the rover goes out and brings samples back to the lander where the spectroscope actually does its work. Or you want to grab samples of air, doing some upper atmosphere study, or in the case of our underwater vehicle, you want to go out and grab water samples and bring them back to the lab because you want to do some DNA analysis on the microbes that you find in the water. So this is broadly in the physical sciences called the problem of ex situ sampling. The problem is you want to bring back physical samples, but they can only be labeled offline in batches. So you want to know what microbes are in the water here. We're not quite at the stage where you can send your sequencer out on this machine to bring water samples back to the lab, and then you do some sequence analysis, right? So in a computer science setting, all the labeling must happen offline and in batches. And it turns out there are lots of interesting domains where people do this, right? So we want to be able to automate this. We want to be able to automate this to some extent. You can very quickly see that ex situ sampling problems are dominated by the question of where to sample. But remember, in this talk, when I say sample, I mean gathering a physical sample. I do not mean taking a picture or gathering a piece of data. I mean taking a physical sample. And there'll be some ramifications to that. Okay? All right, so I particularly care about this water domain. I particularly care about taking water samples. Microbial life in the water is a whole world unto itself. It's a fascinating domain. I know very little about it, but I've been educated to some extent by my collaborators. And here's a satellite view, right? So this begins on the 19th of September and goes all the way till the 8th of October. We're looking at Monterey Bay on the western coast of the United States in a satellite view, and it's colored, and what the yellow shows is the chlorophyll concentration in the water. And you can see, chlorophyll is found in algae, and so it's a good signal for the biomass, right? And you can see over time, it sort of moves, it advexes around, the algae grow, they shrink, they get eaten by other algae, all kinds of interesting stuff happens. And usually, if they release toxin in the water, then people put up signs like this, right? And some of you may have seen signs like this, right? Often these signs go up after all the bad stuff has happened. So what this really means is you shouldn't have gone in the water yesterday, which is not particularly useful, right? Because the sign wasn't there yesterday. We'd like to change that state of affairs. So that's where the sort of the impact of the work, I think, will be felt. Today what happens is if you don't catch this in time, stuff like this happens. But this is an actual picture from Redondo Beach, not far from USC where I work. One day, in the harbor, there was a dreadful smell, and when people looked, they saw that there was a algal bloom just offshore that just killed a very large number of fish. Right? So it can be a dramatic problem. Okay, so now on to robots, right? So ocean robots are some of you have worked with them, so you know this, but for those of you who haven't, they're actually a commoditized thing. You can go buy one, right? You can buy incredible machines that you can in fact deploy straight into the ocean. This was not the case 30 years ago, but due to some fairly steady progress in both the mechanical infrastructure, some of the algorithms for doing underwater localization, and also some other techniques like better computers and better batteries, you can buy a lot of vehicles like this. This is the vehicle at the Monterey Bay Aquarium Research Institute with the people we collaborated with, the Dorado vehicle. This is a commercially available glider vehicle, which is a buoyancy-driven vehicle. We deploy a fleet of 4 of these in our laboratory. By the way, it doesn't always have a string attached to it at the back. This is just so my student could film it while it was in the ocean. I mean, normally the whole point is not to have it tethered. And what these vehicles come with is, you buy them, they come with a certain amount of autonomy. They come with localization. They come with waypoint navigation. You can buy a vehicle like this today and actually give it a set of GPS waypoints, go throw it in the ocean and say, go to this waypoint, dive, go to this next waypoint, dive, go to this next waypoint. And these vehicles do a fairly admirable job of doing this. Now, to all of you who are under 25, you're a little bored. But the point is, this is quite cool. This was not how things were when I began in robotics. It just wasn't. Right? It is remarkably cool. But what it means to the roboticist is, what next, right? What should we do next? It's kind of dissatisfying to have autonomy be this thing where you give a vehicle a set of waypoints. In 1980, I'd have killed for a vehicle like that. Today, I feel like, that's a very low piece of the stack. I need to move up on this food chain, right? And so what's missing, obviously, are the kinds of things that many of you work on in many areas of robotics. These vehicles lack the ability to interact with humans in some kind of sophisticated way. I want to give these vehicles a more sophisticated notion of a mission, other than just a set of GPS waypoints, right? I want them to do more onboard decision making, want to prime them for long duration autonomy, and then ultimately the holy grail is for them to be able to do physical interaction of the environment. My talk today will not talk about physical interaction, though we have some new work beginning on that. We'll talk a little bit about how to change this state of affairs, right? Okay, so here's the problem. We want to be able to send this vehicle out into the ocean. The vehicle measures environmental parameters. already sort of is good at doing that. You can measure things like the temperature, the salinity of the water. You can measure various salts that are dissolved in the water, the pressure, all kinds of physical and some simple chemical things, right? And usually what happens now is this vehicle is sent out into the world. It has a bunch of canisters on board. These are small syringes. So based on some logic, either based on some hard-coded logic or there's some simple decision making, these canisters are filled with water. These syringes draw water from the ocean. Each syringe is brought back to the lab where a smiling lab technician does some analysis on them. And what you get out is a picture that looks like this, right? So here's different species of zooplankton, and here's their abundance. And so this creature was pretty prevalent in the water sample, right? As far as we're concerned, that's what we learned from this. You get a fingerprint of the water sample you brought back. And of course, if you study marine microbiology, you're interested in why certain creatures do better than others and so on. But in our case, for purposes of this talk, this is what we need to know, right? Now, ideally, what you really want to be able to do is to be able to, while you're in the water, you want to be able to take samples that are predictive of the kinds of biology you can get. This would be quite nice, right? Now, of course, if somebody could cram a giant DNA machine onto this vehicle, then life would be good, but we can't quite do that, right? What we have are proxy measurements. And to many of you, you can see where this is going. It suggests the possibility that if you can learn a model from what you can measure to what you actually want to sample, then life could be quite nice. You could actually tell the vehicle, bring me water samples that look like this, and the vehicle could go out and sample. And so rather than say, go to location XY, you could, and hope that the biology is what it is, you could in fact say, bring me water samples that look like this. And that's the kind of thing that I mean by moving up on the autonomy stack, right? People don't like to give GPS coordinates to vehicles like this. They would much rather command what is needed, right? And so that's, I think, kind of cool, right? So here's the sketch of the overall ecosystem. What we're going to do is we're going to begin by assuming that the hard work of the people in the previous decade, where they have got these labeled samples, is something we can harness. We will use these as training data. We don't have a large number of these, and our paper gives some results on how many you need, but we use these as training data. We will learn from this a forward model. It's a simple regression model of how the physical parameters predict the biology. Based on that, we're not done because there are only 10 syringes on the robot. So even if you can predict the biology, I want the best 10 places to sample. And so we will design a utility function designed in some way so that you're going to get these best ones. And I'll show you how to do that in an online fashion. You bring those water samples back, you do lab analysis, and then if you like, you can add that to your training data set and you repeat this. So hopefully over time, you develop a vehicle that's good at doing this, to which you can simply show a pattern like this and say, bring me water samples that are hot for alga X, and the vehicle will go out and do it, right? That's the idea. Okay, and so you can see that there are a few pieces here, right? We need to build a forward model. We'll build a simple regression model. We need to do some optimization, which is sequential, because as the vehicle moves through the water, we're going to get these samples, and we have to do this in an online fashion, right? So this sits at the intersection of three areas. Many of you probably work in these connected areas. These are quite important in many areas of robotics. Okay, so here's how we set up the problem. Imagine some feature vector that codes for the environment. This is typically a low dimensional vector. D is like dimension 8 or so, 6 or 8. It has things like temperature, salinity, pressure, and things like that in it. We'll call that Z. That's what you can actually measure on the vehicle as you go through the water. There's some training data set that pairs previous Z's with some histograms of organism abundance. I will call those B for biology. So you have some ZB pairs. You don't need a very large number of these to bootstrap, which is a good thing. We will then build a simple model that predicts B from G, and in our case, this will be a Gaussian process. This is not difficult to do from data. You can do it out of the box fairly easily. And then the vehicle has to go out and sample at some locations, and we will show what's a good utility function and how to use this that maximize the utility in an online fashion, right? Using a utility function that I will tell you in a second, right? So the first part is the easiest part. how you build this regression model. There's a straightforward Gaussian process model with a squared exponential kernel. There's a little bit of finesse needed to pick the hyperparameters for this model. In this talk, I don't want to focus on that. If you're interested, read the paper. There's some finesse to how you do that. There's some sensitivity analysis to pick those correctly. But it's not particularly interesting. It's a detail, in my view. So we build a Gaussian process model that stochastically predicts the biology from the physical measurements. Okay? Easy. And we train this using pilot data. Okay. Now, what to do with this, right? Then the thing to do is to somehow now be able to put a utility on a set of physical measurements you see. So you have a model. Now some new inputs arrive, Z. What utility do they have, right? For this, we exploit a technique that some of you have seen and have used. We exploit a technique called the upper confidence bound technique for Gaussian processes, which tries to have the best of both worlds, right? If your model is good, then perhaps all you need to do is to maximize the mean, right? You simply exploit the model. If you believe your model is not good, then probably the right thing to do is to encourage some exploration. People have studied this. This is a well-known bandit problem, right? So in this bandit setting, people have shown that the GPU-CB, where you maximize a function of the mean and sigma, produces provable regret bounds, right? So you can, in fact, if you do this, you can show that provably you're doing the right balance between exploration and exploitation. So we just harness this, right? We harness this, I've written it out here. There's a slight subtlety, which is our samples are labeled in batches of K. They're not labeled as they come in. So you have to do this in a somewhat non-standard way. This is a detail. So far, so good. Build a GP regression from pilot data and trade off mean and variance in a fairly well understood way. Okay. The real problem comes now. The real problem is the vehicle is moving through the water. And by the way, just to give you some context, the vehicle is typically deployed on a mission where it's doing a lot of different things. So usually people like me don't have complete control of the vehicle. Our problem is, given a trajectory that some other people have planned for the vehicle, along the way, which 10 samples would you take? So I don't have complete freedom to say, oh, I changed my mind, let's go back over there, and I changed my mind, let's go over there. Someday I will have that freedom, but not quite yet, right? So I will show you a few pictures like this, so let's take a second to understand what these pictures show. On the x-axis here is time. On the y-axis here is the depth of the robot. And in any oceanography picture, zero depth is at the top, and deeper you go, goes bottom. So this is the vehicle at the top, at the water, just at the surface, and all these low points are where the vehicle is dived. And so what I'm looking at is the vehicle diving and coming back up and diving and coming back. I'm not showing you how it's moving in x, y. Obviously, it's not just bobbing up and down in one place, but I'm just showing the variation with depth, right? And what I've false colored onto it is where there is a lot of chlorophyll in the water, so where the microbes are. Okay? Underneath is a picture where this axis is time and it's aligned with this axis, and on the y-axis I'm plotting the utility function, this UCB function. And you can see it has a bunch of peaks. There's a bunch of high utility points, right? They all look pretty good. This is a good point, and this is a good point, and this is a good point. There's lots of places where this utility function spikes, at least according to the model that this GP had at the time. Right? So where to trigger the actual sampling event? So when the vehicle arrives at this location in time, it sees this peak. Should it in fact take a water sample or not? That's the online problem that the vehicle has.
So why is there a limitation in the number of samples?
It's purely physical. The vehicle can carry 10 syringes of a certain capacity. Bigger vehicles will carry 100. But I will soon show you how many possible sampling locations are. So the number of samples is small relative to the overall number. And that's why the problem is interesting. Right? So that's a good question. I said at the beginning of the talk that when I say sample here, I mean physical sample. This is one place where physical samples differ from taking pictures. If you're really old, once upon a time taking pictures was expensive too. The disk would fill up. But that is just not the way we think today. But this is not information directly extracted from the environment, taking a physical sample, right? So there's a limitation. Okay, so now where to sample, right? By the way, one question that's worth answering is you could say, this doesn't seem like a very hard problem because all the biology seems to always be at the same place. Look, all you do is sample at 5-meter depth. I mean, after all, it's clearly there's no point in sampling here. There's none of your favorite microbes live there. So why not just, it seems like you're always red in roughly the same depth. I just want to remind you that I'm not interested in just sampling any old alga. I want to sample where it's going to be hot for one species. And they all like to live in the same place. That's exactly why the problem is hard, right? And by the way, it's not surprising that all the biology is here. If you just think about the ocean a little bit, these are plants in the water. They need sunlight. The sunlight comes from the top. The nutrients come from the bottom. These animals have figured out the right balance. That's why they all live at the same depth. This is beautiful, right? I mean, so that's interesting. Okay, so our problem is you have K syringes and you want to fill them up with water. How do you choose K samples to maximize the sum of utility from these K samples? I will choose an additive setting for the problem here. You can think of other settings, but the additive one is the simplest one, right? And so time, depth, you're swimming along, where to trigger these K samples? Maybe these are the right K. How do we find them? Okay, so there are 3 basic challenges, right? The first challenge is if you are too greedy in picking these samples, then you will miss out on future hotspots, right? So you say, the utility is super high, let me sample. You do that 10 times and now your syringes are contaminated. This is the other problem with physical sampling. You don't undo, right? So now this contaminated syringe, you can't push it out back into the ocean and take a new sample. It's essentially contaminated, right? The other is if you're too conservative, where around the next bend will always be the hot spot, you can return with too few samples, which would be a tragedy, right? And the really annoying thing is, it would be nice to do this without setting thresholds. It would be nice to do this without being ad hoc. You know, I can say, oh, you have this utility function, anything of, you know, if it's on a scale of 1 to 100, anything about 90 is great. Yeah, but I don't quite want to do that. I want to do something a little more principled. Okay, so the question is, can we? And as is customary in academic talk, the answer is yes, we can. Otherwise, I wouldn't be saying it. So it turns out that some very smart people studied this problem. This goes under the name of optimal stopping theory, right? And some of you may have studied this, right? People have thought about this. So there is an old problem called the hiring problem, which is an abstraction of this setting for the one sample case. Imagine you want to hire a person to fill a job. And because this is a somewhat stylized setting, there are N candidates. They arrive for interviews sequentially, and they arrive, and they're IID, so they're not related to each other. They're not correlated with each other in some way, right? Your goal is to choose the best candidate online, which is sort of not quite how we do hiring, but okay, for the purposes of this. And the decision is irrevocable. This means when a candidate come in, you interview them, you rank them based on whatever your metric is for ranking them, and you make a decision on hire or no hire. And that decision is irrevocable. If you say no hire, they go, you see the next candidate. If you say hire, the process stops. In this setting, how should you hire? And it turns out there's a crisp answer to the question. You should observe the first N over E candidates, which is about 1/3 of them, and you should hire the next best. If you do this, and by the way, if there's no better candidate, you hire the last person. If you do this, you can show that the probability of choosing the best candidate is exactly 1 over E. It's a well-known result, right? Now, you may say that, well, that's not great, but I argue that it is far better than saying, notice that there is no threshold on the utility function. You don't say candidates above 80 on a scale of 1 to 100 are okay. Nowhere do you have to do this. You only have to compare people, right? It also takes no position on how you pose the utility function. So this is independent of your utility function, as long as you consistently apply it to every candidate, right? So I think this is a kind of a neat way to think about the problem. Our problem is a little more challenging because we have to select effectively K candidates online. Each water sample is filling a slot, so we have to pick K of these online, right? This problem is interesting. You have to pick K candidates online, and by best K, I mean in the additive sense, which means I want the best sum of their ratings, right? How do you solve this problem? Well, it turns out that this too, something is known about. What you should do is you should split your hiring window into K segments, And in each segment, you should apply the algorithm of the previous section. Okay? And it is known that if you do this, you will get a guaranteed competitive ratio of at least this. Okay? And then some of you will look at this and say, that's awful. That's a factor of 20. I mean, generally when you show a competitive ratio, it's like 2 or 3. I'm saying 0.05. So that's not great in itself, right? Which suggests probably that based on our experiments at least, suggests that the analysis could get better, because the competitive ratios we get when we actually do this in practice are nowhere near as bad as this. So in our minds, we will think of this as very loose upper bound, but it's still lower bound, but still preserves this property that you don't have to actually fiddle with the utility function. In other words, my biologist friends can pick the utility function. I don't really care what it is, right? So this is kind of a nice property. By the way, this problem is hard because n typically is 1,000 times bigger than k in our setting. So n choose k is a huge number. That's why it's worth doing. I mean, if that number is not big, you don't need all these gymnastics, right? So roughly speaking, it's a factor of 1,000, right? I'll show you some data in a second. That's one. The other is the answer to the best k candidates is not the same as picking the best k minus 1 and adding 1 to it. Correct? If you think carefully about it, it is not the case that you can solve the 9 problem and get the solution to the 10 problem by independently solving the 1 problem and the 9 problem. Right? So this is a submodular selection problem. So it's a difficult problem. And the combinatorics are not in your favor. Right? This just says what I said in pseudocode. I will skip this. This is not important. So now back to our workflow, right? So you build a model from some training data. You use a UCB utility function. But the more interesting piece is you design this online best choice technique, which maximizes the sum of the utilities of the k best samples, right? And those are the places you sample. So let me show you some data. Let me show you what actually happens. Yeah.
Can you give some intuition on why is it not additive? Why can't you just solve the 9 problem in the 1?
Yeah, I mean, you can think about it as follows, right? It would be if you were allowed to go back. So, you know, if you want to pick the best 10 samples, right, then depending on the distribution of the samples, you may pick one in the beginning and one at the end and somewhere in the middle, etc., right? If you already pick the best 9, right, then what you have left to you, because this is an online setting, you can only pick the last one from a limited set. If you were allowed to pick the last one from everything you had seen before, life would be much better. But because this is online, your commit cannot be reversed. And so you either solve the 10 problem or you solve the 9 problem, but you can't actually say, I'll solve the 1 problem at each step.
Just because you can't travel back.
You can't travel back,
okay.
Right? So let me show you some data of how this was done. So we do this in a couple of different ways. First, we're going to mine some old data and essentially make the vehicle just fly through some old data. So all we do are emulate these sampling runs. The data are real, but we are essentially faking the vehicle traversal. Then I will show you some real data from the ocean where the vehicle is actually in the ocean and doing this online, right? And so we took some old data from old surveys. This is the Dorado vehicle. I showed you a picture of it. What you log are the physical parameters. And this one biological parameter, which is the proxy for the biomass. This is the fluorescence. And the goal is to acquire these simulated gulps of high abundance phytoplankton samples. And so here's how you do it. On the first couple of days, you run your pilot surveys with random sampling. There's no model in the beginning. You learn the initial GP model, right? Once you have that, you run some targeted sampling using the learned model. The result of that allows you to update the GP model. Then you repeat the process, right? You update the model. And you do this throughout the 17 days worth of data you have, right? All the data are cached. This is just old data. We're just exploiting it. We call this exercise 1 campaign. We're going to repeat this campaign 100 times with random restarts, okay? And each time we are interested in sampling, we're filling 10 canisters out of 45,000 potential locations where we could have sampled. That's about a factor of 1,000, right? So you can see the density at which you need to sample is very, very low. It's very, very low. So theoretically, the number of choices is 45,000 choose 10, which is a silly big number. Right. Okay. These are sometimes interesting if you sort of care about what the model looks like. It's worth visualizing, so let's spend a little bit of time on this. Once again, this is my picture where I show time on this axis and depth on this axis. The vehicle is sort of navigating through this. It's a little hard to see, but these plus signs are where the vehicle actually chose to take samples. You can see that some of these gulps are at the ends of these K segments. For lack of better candidates, we're segmenting the window into K samples. There are some gulps within segments, right? You can see the biology we predict and the actual biology here. We have all the old data here, so we can overlay the two. A little bit of a look at what the abundance model looks like. So this is a little hard to understand, but on this axis is the size of the particles the vehicle sees in the water, called backscatter. It's an optical instrument that measures the sizes of the particles. And this is a temperature axis. This is temperature in Celsius. And what you're seeing here is the mean of the model, basically. Generally speaking, we don't tend to visualize these pictures too much, so may not mean a whole lot to you because I'm sort of skipping over the details of which species this is and so on, but just to give you some idea of what these models look like. You can see the models refine over time, so here's what things look like on day 1. So the top 2 panels are the mean of the model, the bottom 2 models are the variance. On the right is the UCB strategy. On the left is just a mean-driven strategy. You want to focus on this one because it's going to end up being better than this one. As you can see by survey 4, the model is much more focused. By survey 16, the model is significantly more focused using our technique. And so it grabs onto the areas where the high abundance samples are likely to be found. The model is higher dimension than this. This is just temperature versus backscatter. There's also nitrate concentration and pressure and so on. There are about 8 dimensions, but I just picked one slice through it. If you read our paper, we talk a little bit about what input parameters we're more sensitive to than others, and in fact, we don't need to build the entire GP. We can only build pieces of it, so there's some details which I've not talked about here. We're actually most sensitive to temperature and backscatter, which is the model I showed you. So that's all good. Let's look at some, and since I told you that it would have the lowest regret, I'm just showing you a picture of the cumulative regret. So you add the utilities of the samples you took, you add the utilities of the best 10 samples you could have taken, you take the difference between the two, this is the cumulative regret. You want this number to be as low as possible. Right? And with increased surveys, the regret is going to grow. You want a technique where the regret grows as slowly as possible. So I've compared several things here. The technique in black is the one I've been describing to you, which is labeled all GPUCB. All simply means we don't throw away any data. There are other ways of doing it. There's a variance-driven technique with all the data. There's a mean-driven technique with all the data. There's a window technique with UCB where you throw away older data, but you still do the UCB technique. And then there are all kinds of other ways of doing it. And of course, you can just do this randomly as well, which is kind of the straw man. You should never do worse than that. So that just gives you an idea that we do pretty well.
Question. I don't know what your policy is. We can interrupt you in the middle.
Yeah, please.
That's great. I know. I was trying to be polite. Upper confidence bound is one measure. I was curious, see the question I asked, what if you use expected improvement?
We have some, we've done it on a few of our data sets. On our data sets, actually, we don't see a tremendous difference.
Right.
And that could be partly because even though, so our data are a little bit strange, right? They're all gathered from ship surveys. So even though in theory you're looking at XYZ, the ship describes a particular trajectory through the space. So actually we're looking at a lower dimension projection of what could be. So we are probably not seeing as much variation between some of these techniques as you might expect, because we're not truly exploring every spot. We're only looking along a trajectory.
Yeah, let's ask about that in a second. But so the thing I find interesting is if you had just gone to maximize median or variance or some combination, you would get a worse answer than you're getting.
Yeah.
So I'm just trying to figure out when does being smart start to?
Yeah, so one of our challenges is that you could sort of, you're being very polite, but you could say, there's not a whole lot of difference between these, right? I mean, being smart didn't seem to buy me that much. Like I could have been, clearly all of these are smart enough compared to that, right? But there's very little to tease them apart. We're not sure. We're not sure whether you need to, in fact, it's pretty clear that even windowed UCB techniques will do just fine. There's not a whole lot to choose among.
And I wasn't trying to be intrusive, I was because in what we do,
yeah.
we do a similar thing where, and sometimes random does as well.
Yeah.
And that's really sad.
Yeah. We feel like targeted sampling has certainly bought us something. I'll not go so far as to say that you need to be, that we need, I don't think we're going to spend a whole lot of time trying to get further refinement to this. Our main interest now is marrying this with planning techniques so that in fact we can simultaneously do trajectory optimization with sampling.
Right,
and I think that's where we are really going to get some payoff.
And I guess that's the magic is. I was just curious that is there a fundamental principle here that says all these are almost more or less the same. So excuse me, there's a quantifiable benefit and the fact that they're more or less the same hints at that.
Maybe. I don't know.
Okay. And since I interrupted you already, are you? I realize these are ships. You're not steering the boats.
Yep.
Or the vessels as they
yep.
call them. Are you going to talk about later on that what happens if you can aim?
Not in this talk. We have some work now that's happening on trajectory optimization along with this, but we don't quite have prime time results from that yet.
Right. Will be on big ships.
It'll be on big ships. Yeah.
You're steering
yeah.
big ships.
Yeah.
Oh, that's great.
Oh. Question.
Well, since this is fairly well stalled, so you keep saying just taking the sum. Are you really just adding the
utility values? Yeah.
No penalty for sampling immediately adjacent points?
You don't need a penalty, right? Because our sampling policy spaces the points out automatically. K samples means they must be in K segments.
Right.
And so there's no need for any other penalty term. By the structure of the algorithm itself, they will be in K separate windows, non-overlapping windows.
Right. The theory you're building on, that's just a random side effect.
Yeah, it's not clear whether it's a random side effect or whether it's actually a good thing to do, and in fact it's possible to prove a tighter bound for that setting. So you're right. It could be that there are better ways of doing this, and then better bounds will follow, or maybe that's a very sort of worst case way of doing it, and the bound is in fact tight. In practice, we do far better than that regret bound, far better. So there's a clear gap. I can't quite say whether the gap is because the sampling strategy isn't quite right or whether the analysis isn't tight.
Sorry, extension to the same question. You say the utility is additive, but I see submodular secretary here, so where does the submodularity of it comes in?
The submodularity is just in the subset selection problem. So you're picking these subsets, and it's the point simply is that you can't pick the subset as the sum of two smaller sets. So the submodular secretary setting is this K sample setting, where you can't pick the solution to K as being the solution to K minus 1 and the solution to 1.
Okay.
But our regret bounds are all on the sum of the utilities. So the cumulative regret is just measured by adding the utilities and subtracting from the total number.
Not a practical issue.
Yeah.
I like the attraction to the iron problem, but if you're doing this, can you actually measure the utility as you're going along?
Yeah. Absolutely. Very good question. So very pragmatic question. Physical measurements coming in. Is there something that stops you from actually computing the utility? No. So we can compute the utility in real time. It's a simple function before you take the sample. And so that is essentially instantaneous. Because remember, you're only relearning the GP after every traverse. So there's no online learning going on. That's all happening in batch. So all you're doing is computing a mean and a variance and then a simple combination of the two. And you can do that. You have an existing model, you take physical data, and you can do that. Take about 1 sample per second. You can update the utility value every second, roughly. Yeah. It's a good question. Okay. Now, this is really annoying. It's really annoying. Okay, there's a picture of a ship here, okay, which is missing somehow. Okay, maybe at the end I'll find some way to show it to you. But there's a beautiful picture here at sunrise, imagine it, of us on a ship with our vehicle. But Let me show you the data. It's really annoying. My only ship picture. Okay, so here's a field trial. Here's a field trial. Here's some data from a field trial, right? So here, there's a very specific goal. Before the field trial, the vehicle is told to acquire high abundance samples in one particular species of alga. This species is called Pseudonychia, PN. It's found in the waters of the western United States. It's potentially toxinogenic, so people in microbiology care about it. It can do harm. So we have some old data that allow us to learn our model. That's how we bootstrap. We do some work to pick the input variables, and the GP is a little different than the one I showed you, but it's not quite important for this talk. And then you go out in North Monterey Bay on the Dorado vehicle to acquire 9 samples. And I believe the day we did this first run, one of these sampling syringes was not functional, so we only gathered 9 that day. Okay? So here's our trained model. Here's the prediction, and here's the variance that goes with it. Again, this is temperature versus fluorescence. Fluorescence just shows you how much chlorophyll there is in the water. So you can sort of just get, I mean, this is just one way of looking at this phase space in 2 dimensions of what the picture looks like. Here are the samples we actually acquired, right? The pluses show you where the vehicle was on its run, simply chose to take samples. Right? So we give this vehicle this goal that you're going to sample where it's hot for Pseudonychia. We give it the initial GP model. It implements this online best choice technique, comes back with samples that were taken at these locations. It does 2 things. It comes back with these samples, and along with each of these samples, the vehicle also has an estimate of the abundance of this species at those locations based on the model it has. So it commits. It says, I sampled at these locations, and I predict that at this location the abundance is going to be so much, at this location the abundance is going to be so much, etc. Right? That's the deliverable. Okay? So what you do is then you hand those water samples off to the people who go off and do the ex situ analysis. So somebody goes with those samples off to the lab, and they actually try and figure out how many of these species were in a unit volume of water. Right? And usually they do this optically. There are some DNA methods as well, but generally this is done optically. Right? By looking, these algae have different shapes, so somebody actually looks at them under the microscope and does cell counts. That's typically how it's done. Right? So here's the predicted value of the abundance on this axis against which syringe was used. So you can see our technique predicts that the first syringe will contain a fairly high abundance, and the second one even higher, the third one even higher. The units don't quite matter here. We were also told to take at least 1 water sample in a location where the sample was not going to be hot. And this is generally good practice, so that if you return with all samples hot, it may be the case that the whole ocean was hot for the alga that day, and it's not clear how you separate the 2. So generally you run these missions where at least there's 1 baseline sample where the sample is not hot. So here's our prediction, right? So we can produce this picture along with the water samples. So basically what we do is we hand this picture to the people doing the analysis and say, we predict this is what you will see, right? And here's what they saw. It's a little washed out. I will overlay our predictions on what they measured, right? So if you look what they measured, there's some deviations. Like we got number 3 wrong. They measured significantly higher than what we predicted. But you can see very broadly that where we predicted there would be almost nothing, there is almost nothing. This one particular species, where we predicted high concentrations, almost invariably there are very high concentrations. Okay? It's pretty nifty because it's all being done by people like me who really don't know anything about microbes, right? So we can really say, we can tell you what's going to be in this water. It's going to be hot for exactly the species you want. Right? We've done many runs. So this is our first one. This is the one I'm most proud of. It's not actually our best data set, but it's the very first time we were able to put this on a vehicle. It went to sea. It comes back. You make the prediction, you hold your breath, you actually see whether the measurements in fact line up, and when they do, it's quite nice to see. Right? And so I want to sum up here a little bit. What I want you to take from this talk is that this is really an exercise in doing stochastic online sampling with constraints. Right? The constraint is you can only take so many, that they are irrevocable. You will notice that our model that we build is not a model of the geography of the ocean. So it doesn't say that this species is found at this location. What it says is that this species has a particular ecological niche, which is a certain kind of temperature, a certain kind of pressure, a certain kind of concentration of minerals in the water, and that's the ecological niche where you will find this species. That's not entirely surprising. Right? And so as the water moves around, you don't necessarily lose anything. We're not building models of where you will find things. And these kinds of plants don't live in a particular place. Right? This is the first data-driven experiment of this type in marine robotics. It's kind of cool to be able to do this. Right? And I think most importantly to somebody who does robotics, I think it allows people who use vehicles as commodities to task them at a higher level. My biology friends can now say things like, bring me that microbe. You can throw a vehicle into the water and say, I care about this microbe, go get me that microbe. Other than saying, go to this GPS location or go to that GPS location. And to me, that's the important thing about robotics, right? Autonomy means being able to leave the robot alone and not joystick it, right? It's very important to be able to task it at a high level. So this is one small contribution towards that end. That excites me more than any of the other stuff associated with it. And it's kind of cool because people have been sampling for algae in the water since the early 20th century. This is a slide taken from NOAA 100 years ago. People going out with plankton tow nets, gathering them up, bring them back and looking at plankton under a microscope. And today we do it like this, which is pretty cool, thanks to the magic of robots. And I think that's a really cool thing. You can really sort of open up the underwater domain in an interesting way. So I'll just stop. I'll acknowledge JD, who is right here, who was the lead student who worked on this. Several other people collaborated and helped. My collaborator at Mbari was Kanna Rajan, and some collaborators at USC. And I'd like to thank ONR and NOAA for their sponsorship. I'll now stop here.
The way this goes is we ask questions now. I think you're all used to that. The only thing is Gaurav has one more meeting after the seminar, so we'll take a couple of questions and then I'll rudely stop you all, if that's okay. So Kaushal, please.
So the microbe concentration changes with respect to time.
Yeah.
Can this technique still be applied? So is it just changing the GP model?
Nope, you don't change the model. Because remember, the model simply maps the physical parameters you measure to the utility.
So it's only initial, right? So
No, the temperature is one of the inputs. Temperature is one of the inputs. Time doesn't make any difference because if the temperature changes over time or if the pressure changes over time, when you say that these microbes change over time, there are only three things that are happening. One is they're being physically advected by the ocean. That's why they change in time. Two is they multiply. Three is they get eaten. But there's no other time-dependent effect on these microbes, right? And so there's no particular value to the time. There's nothing like they'll be here and during the day and they won't be here at night, right? So it's not time dependence in the way I think you're thinking. It's time dependent in the way that if you observe a certain patch of ocean and ask whether the microbe is there now, and then whether it's there 10 seconds later and whether it's there 100 seconds later, you'll get variation in time. But that doesn't affect the GP at all, and it doesn't affect your sampling strategy at all. Does that make sense?
I noticed that you're trying to somehow or other, your definition of utility seems to be to get it at the center of the microbes habitat.
Yeah.
Was there any effort made to try to find the edges of the
No, it's a good question. It's a good question. We haven't actually figured out. So now this opens up lots of interesting questions, right?
Where does it drop off? Where does it stop happening at the bottom?
Yeah, so the edges are, it's species dependent, and so it's quite a bit different for different species. So some people have suggested to us that we use these techniques to figure out these bands, so characterize entire habitats using them. We haven't done any of that. It's a different question. It's a very interesting question. Yeah.
So if I understood right, you divide your sampling area up into K bits of power many, and your algorithm will take one sample per.
Correct.
So is there any way you can figure out how many or how few bins you need, or?
The number of bins, under the assumption that you want to fill each syringe, the number of bins is equal to the number of syringes.
Right, but you need a machine with 10 syringes, or can you use one that's cheaper that only has 4?
If you, it really depends on how many samples you feel are worth collecting. Generally, biologists tend to want as many as possible, so it's a.
Just wondering if like there's a function between samples taken and your maximum regret or something.
Yeah, I don't know that that's the case. I mean, the cumulative regret will always grow over time because you'll never quite get the. So in fact, it only gets worse with every sample you take. You accumulate more cumulative regret. Yeah.
So if you have a region where you're, let's say, just on the surface going for a long time, so you would have to take a sample. But if you...
Yeah, so it's a good question. This is piggybacked on the idea that the trajectory design problem is decoupled from the sampling problem, which it is not in reality, right? So nobody asked me saying, who told the vehicle to dive and then come back up and then dive and then come back up? And so in fact, what we work on now, we'll have some results on this soon that I think will be good, is to simultaneously optimize for the problem of navigation and sampling, right? Which is when to dive and when to come up. Every time you dive, your localization degrades, and every time you come back up to the surface, you get a GPS fix. So actually, if you consider the uncertainty in pose as part of your cost, then in fact the problem is a little bit more general than this, and we have some results on that as well. And so we're working on some. But here the assumption was that you picked a set of dive profiles and you picked an XY trajectory, and now you just impose a sampling pattern onto that. But it's a limited view of the problem.
I think staying even with the independent assumption that trajectory and sampling are two independent problems.
Yeah.
In this policy, you'd still end up sampling a region where you don't have anything at all just because you're, I don't know what you call it.
The technique said you must. Yes, exactly. That's exactly the thing, right? So if there happened to be a region which was completely bare, then the algorithm would guide you to the last sample in that region and you'd be forced to take it. Yes. So there's always this potential of doing that. Mm-hmm. Yeah. Which suggests that there are better techniques. Right. Yeah. And remember, it's only because it's online that it's tricky. I mean, if it was batch, there's nothing to it, right? I mean, they are uninteresting problem. I mean, the combinatorics are still hard, but there's nothing intrinsically that difficult. Then you can solve it, right? Yeah.
Because also the thing is that the hiring problem assumes that all the n samples are going to come from the same distribution.
Yeah.
Here, if you are going to take a trajectory through the ocean, you know that some regions are going to be less attractive and some are going to be more attractive.
So that's a very good question. Sometimes I get asked this saying, so the hiring problem, the solution is that, the distribution is IID. But there's nothing particularly IID about this. I mean, you're swimming through the water. The next piece of water looks exactly like this piece of water. So anything but IID. But here's where the sparsification induced by our technique helps. Because of the windowed approach, you naturally will sample in disjoint windows. And so you get that. But that's not I think there's room for improvement there. There's definitely room for improvement there. Yeah, it's a good question. Yeah.
So one thing that wasn't clear to me is like, are these windows fixed length? You just like divide the trajectory into fixed lengths?
They're fixed length, yeah.
I mean, you can just use the prior information and look at what your GP looks like and decide on the window length.
Can't do it. Can't do it. The prior tells you the mapping from the GP to the utility values. But before I go into the water, I have no idea what the salt concentration at a particular location is going to look like. So I actually cannot use the model until I'm in the water. Do you see the problem? I can only instantiate the model when I make the measurements. And I can only update the model when I'm offline. That's exactly the problem.
Your utility is mostly a classification rather than a regression, actually.
Yeah, sure. I mean,
that's how
I mean, I consider it a regression on the samples, and then you're predicting, you're computing the utility value when you see the new inputs. Maybe it's a stretch to call that prediction, but you just compute the utility value. Yeah. Good question.
This will be our last question.
So I was just going to go off of that. I was thinking that too. Do the gliders ever travel in areas where there's going to be a whole patch where it's just outside the range of that? Or is that just...
No, so you guys are asking good questions, right? So I'm pretending as though actually you don't, you know a few more things, right? You have satellite imagery that can track where these patches are, and you can choose to live within these patches. We actually have a paper on tracking these patches. And what you do is typically, in fact, the survey that these vehicles run, all of this survey is contained inside one of these hot patches. So it's in fact not as unconstrained as I said, where you just pick some random trajectory and you pick samples. What you actually do is use satellite data to say, there's this patch, and using an ocean model, I'm going to predict the patch is going to be here tomorrow. So I'm going to design a trajectory inside that patch, and then we're going to run this technique in there. So there's some other wrappers around it. But good question.
Okay, let's thank our speaker.
Thank you.
All of you can go away.